Dear learners, I welcome you to this module which is related to understanding of RNA and its general structure. This module will help you to create a vision regarding what an RNA molecule is and what kind of modification it undergoes or it can undergo so that it can be used for different and versatile processes inside the first of all, let us recall in one of my lectures on DNA, I had explained about nucleic acids, right? So, this is the second type of nucleic acid. Let's just start that what is a nucleic acid? Nucleic acid is a polymer of nucleotides, and there we had already studied that nucleotides are of two types. One which we have already completed, that is DNA, and the second one which we are now starting, that is RNA. So today we are going to focus upon the second type of nucleotide, that is RNA, and its full form is ribonucleic acid. We can recall many things from DNA. RNA has different structural annotation, that it has a sugar which is ribose. Okay, and on the basis of presence of the sugar, this name ribonucleic acid has been given to RNA. This is one of the major difference between DNA and RNA, that in DNA deoxyribose sugar is present, whereas in RNA ribose sugar is present. Apart from this, RNAs are generally single-stranded molecules. Any kind of RNA, when we study inside a cell, they are single-stranded molecules. But there is only one exception, that in case of RNA viruses, okay, you can go across a double-stranded RNA. So that is one of the exception, as it was in case of DNA, that single-stranded DNAs were encountered in certain viruses. So this is one of the major difference between DNA and RNA, that ribose sugar is present, then it is single-stranded structure, as well as in case of viruses again, RNA acts as a genetic material. Otherwise, genetic material is the field of function of DNA in any kind of cell, whereas in case of viruses we find that it is acting as a genetic material for those viruses which are RNA viruses. So these are some exceptions which we find RNA is involved into. The main function of this molecule, whenever we say RNA, is synthesis of proteins inside the cell. Although apart from protein synthesis, there are a variety of functions in which we find RNA to be involved in, which we will come in later lectures. This lecture is basically focused upon the basic structure of the RNA, which helps in the building of different kinds of RNAs, which are showing a variety of functions inside the cell. So being a nucleic acid, it also satisfies that it is a macromolecule, as DNA was a macromolecule, and since it was a macromolecule, therefore RNA also has high molecular weight. Okay, then one more basic difference you can understand here, that RNA in protein synthesis, when they are involved, they are of three types. Okay, messenger RNA, ribosomal RNA, and transfer RNA. Okay, whereas in case of DNA, there is only one type of DNA. There are no types of DNAs. So that is also one of the major differences between DNA and RNA, if you want to understand in that way. Okay, so the thing which here I want to explain is that always when you write the types of RNAs, mRNA, rRNA, tRNA, you have to always write the M, R or T in small. Okay, it is never written in capital. This the learner should always take. Now we come to the basic structure of RNA. Many things are the backbone of RNA is quite similar to that of DNA, but still we will revise here that the backbone of RNA is consisting of a polymer of nucleotide. Just a moment. Yeah, it is made up of a nucleotide. Let us write over here. It is a nucleotide, right? And this nucleotide, it is a polymer. Okay, so here we find that the nucleotide is again made up of three parts. Alright, the first part is the ribose sugar. Okay, the second part is the nitrogenous base, and the third part is the phosphoric acid or the phosphate. So again the same thing, that it is a polymer, it is a polymer of nucleotides. Now we can be very specific that which kind of nucleotides we are talking about. Ribonucleotides. Okay, so let's again refresh what we had studied in DNA. A ribose sugar is the same sugar which we find in DNA, except that, and we will number the carbon. This is the first carbon, second carbon, third carbon, and fourth carbon, and this is the fifth carbon. Okay, so here is the difference, that in second carbon there is a oxygen present. Okay, and therefore this sugar is called as a ribose sugar. Okay, now this carbon, this hydroxyl group is very important. We will come in the further slides about its significance. The nitrogenous base is again attached to the first carbon as in case of DNA, and the nitrogenous base and the sugar together are called as the nucleoside, and to the fifth carbon here is attached the phosphate group, and once the phosphate group is attached, it is attached to the OH, and therefore this H will be removed, right? So it will be a bond between CH2 and the phosphate PO3. So this complete structure is called as a nucleotide. Which kind of nucleotide? A ribonucleotide, which is present in the RNA. Now such ribonucleotides, they are forming a polymer in this way, and they are joined by the phosphodiester linkage in this way. So these are the ribonucleotides, and they are joined by phosphodiester linkage, similar to that of DNA. Then what the difference is? The difference is the sugar molecule, and that only one strand, that only one strand of the polymer represents an RNA, and there is no other strand. Okay. The next important point which we should remember here is that RNA has varying lengths. Depending upon the type of RNA, depending upon the function which is performed by the RNA, the length of the RNA is found to vary much from one molecule to another molecule. It is a more versatile molecule as compared to DNA. And the nitrogenous bases which are present in the ribonucleotides are exactly similar to that present in the DNA, except uracil, where thymine is not present in RNA and its place is taken by uracil. So here we find that adenine will bind with uracil instead of thymine, as we had studied in case of DNA, and cytosine will bind with guanine, similar as it bind in it binds in DNA, and the number of bonds are also same. Adenine and uracil has double bond, and cytosine and guanine has triple bond. So this is the basic structure, and here we find that the skeleton, although it is made up of the sugar and phosphate molecules similar to that of the DNA, but here the nitrogenous bases are not so much involved in a very regular base pairing because it is a single stranded molecule. Uh, the RNA, as we had already studied that it has a ribose sugar, uh, we find a very typical characteristic of RNA over here, that the RNA has an ability to undergo modifications. Okay. Uh, Modifications can be done by modifications can are very easily done in RNA molecule, and the reason is the second carbon which has the hydroxyl group. Okay, now these modifications are done by various kinds of enzymes. Okay, and the modifications which are done are very simple modifications, like for example addition of CH3 group, which we call as methylation. Okay, now the significance of this methylation is that it helps in formation of different kinds of bonds, and also the second carbon gives an reactive nature to the RNA, which makes it more prone to hydrolysis, and therefore it makes RNA an unstable molecule when we compare it to DNA. So this is the significance of the second carbon hydroxyl group, that it allows modification, it allows hydrolysis, and therefore the RNA molecule is not so stable as compared to the DNA molecule. And the structure of RNA was first of all described by Holle in 1965. So let's just sum up that what is the general structure. We had studied that it is a single stranded molecule, and there are complementary sequences which leads to the intra-chain base pairing. So we find that if an RNA molecule, we are saying that it is a single stranded molecule, so the structure must be like this. Okay, this is the starting point and this is the ending point. But we find that this molecule undergoes a sort of coiling or twisting upon itself, and certain regions have uh, pairing, base pairing within the molecule. So there is only one molecule, still we are finding the base pairing. So this base pairings are nothing but they are the result of the complementary sequences which are present in the molecule. Okay. Now these foldings of RNA, they form bulges and helices, which I had shown right now, and these bondings, they actually bring two nitrogenous bases. Say for example, one nitrogenous base is here and one nitrogenous base is here, and both of them are modified. As I told you that RNA undergoes a very quick modification. They are modified in such a way that this nitrogenous nucleotide forms a bond with this nucleotide. The structure resulting from it will be something like this, where there is a bonding between this and this, right? So let me explain that if here and here there is a bond, then what will happen, this part will come close to here, which will lead to such a type of folded and bulged structure. So basically such kind of intra-chain, they are called as intra-chain because they are present in a single chain. There is no other chain because it is a single stranded molecule. So intra-chain bonds, they help in stabilization of different type of three-dimensional structures which the RNA is present. Okay, so it helps in stabilization. And one more very peculiar thing about its structure is that RNA is found to be associated with certain proteins, and those proteins are called as ribonucleoproteins. That means proteins associated with RNA, they are called as ribonucleoproteins. Now, such ribonucleoproteins show activity which are similar to enzymes, like catalytical activities, and therefore we always say that all proteins are enzymes, all enzymes are proteins, sorry, but some of RNA molecules are also acting as enzymes. So this is due to the ribonucleoproteins. Okay. So this is one of the added